Ever since conception, we've been relying on successful DNA replication for our cells to divide so that we can grow, heal, and replace dead cells. When all is done correctly, we should end up with two identical copies of the DNA molecule per each replication. Sometimes, though, mistakes can occur during the process, creating an array of mutations, which can have either little to no effect, or on the other hand, catastrophic effects such as heritable disorders. Though they're extremely difficult to treat due to their genetic nature, the possibility of gene editing or gene therapy exists for some. Gene therapy aims to fix these genetic problems at their source by adding a corrected copy of the mutated gene to the affected tissue. It's not feasible for every disorder, though, as there's a series of considerations that have to be made before determining whether a disease is really a good candidate for it. To be considered for gene therapy, the disorder must result from a mutation in one or more genes, and therefore it can be fixed by adding more functional genes. We must also understand the source of the disorder as well as understand how it works. This is why gene therapy is feasible for treating something like cystic fibrosis, for example, as adding a normal copy of the gene will fix the problem. And it's actually feasible to deliver the gene to cells of part of the affected tissue, mainly just in the lungs via inhaler. So, what's the source of this disorder? Cystic fibrosis is a heritable disorder affecting the cells of the lungs, pancreas, and the small intestines. Is it caused by a mutation? It's most commonly caused by a 3 base pair deletion mutation in the cystic fibrosis transmembrane regulator gene, which leads to us missing an amino acid and essentially ruining the functioning dynamic of the whole gene. Do we understand its biology? Yes. When the gene is functional, it creates a balance between sodium and chloride ions in and outside of the cell, attracting water into the mucus layer and keeping it moist so that the cilia can remove harmful particles from it, preventing disease. When the protein is mutated, however, it can't transport the chloride ions across the membrane, allowing too much sodium in and not enough chloride out. The sodium causes water to flow back into the cell, drying out the mucus layer so it's too viscous and too sticky for the cilia to remove harmful debris, which often causes infections which augment in severity with time as the sticky mucus builds up. Can it be fixed by adding a normal copy? The thing with this gene is the mutation codes for a non-functional protein channel, but it's not going to prevent another normal protein channel from working, meaning that adding a normal copy of the gene should fix the issue. And it's important to remember that while gene therapy seems promising, there's still a lot of work to be done. It's still relatively new and therefore risky, being used to treat ailments that have no other treatments. With more time and more research, though, the practice of gene therapy will evolve to be more safe and more reliable, hopefully expanding the scope of disorders it can treat.